Hi, everybody. I hope everyone can see me. And I want to thank the organizers, even if they are not here, unfortunately, for organizing this nice, interesting event. I will talk about a very recent work with my former postdoc, Mabel Lizzie Rajandran. She's currently at the University of Birmingham. And I will somewhat follow up on the previous talks, or the two talks we had this morning, namely on concentrate on adhesion, cell-cell adhesion, specific, also mediated through cadherin binding. And so I'm a mathematician, so I'm interested in modeling these kind of effects. And we will adopt the multiscale, an adaptation of the multiscale approach. Has already been used by several of you in this workshop earlier this week. So once at a time. So I will first give you some motivation. Namely, I will tell you a bit about binding through e-cadherins. Then I will recap what the multiscale approach is about and explain how we use it here. Present two basic, what I call basic models, such that don't yet include cadherin binding. And then finally, I will show you also an extended model which does include this effect. And if time permits, but probably not, I will show you a few steps of the way of the upscaling from meso to macroscale. So this is more for the mathematicians here. Right. So just to recap, what is cell adhesion? Not necessarily cell adhesion. This is, so to say, cells sticking to each other or to tissue. As we already heard in several talks, this interaction with the tissue is extremely important, but it is, I would say, better understood than the interaction between cells, how the adhesion happening there. So we will, in this talk, we will mostly concentrate on cell-cell adhesion, kind of ignoring cell-tissue adhesion, although we acknowledge that it's very, very important. So adhesion occurs through cells building junctions, and these junctions occur by connections of so-called cell adhesion molecules. I will just abbreviate them to CAMs. So there are different types of junctions a cell can form. So here you can see what looks like schematically. There are junctions cell can make with tissue. You see them on the bottom, here in the bottom. But what we want to concentrate on here is the cell-cell adhesion. And the main example that I have in mind is the adherent junctions through e-cadherins. So this is when cells bind through so-called e-cadherins, already mentioned in one of the previous talks. And these are the main cell adhesion molecules responsible for keeping cells together. And they have extracellular domain, that's where they connect. And then they also reach into the inside. It was already addressed P120 cadherin. So, and through these cells, these cadherins have connection to cells' actin filaments. So affect, and as you know, actin filaments, so what's happening there is has an effect on cell movements or the lack thereof. If a cell sits, for example, is well connected to other cells in epithelial tissue, or when you have less connection, then the cell has, of course, more possibility to move out. Yeah, so it seems quite important to concentrate on these kind of junctions. Right. So again, this adherence binding occurs through so-called classical cadherins, and the most important representatives are E-cadherin and N-cadherin, and specifically N-cadherin is typical for epithelial cells and cadherins for mesenchymal cells. And important is also that binding and unbinding of cadherins depends on the presence of calcium. So from this comes also the name of this cell adhesion molecule Ca from calcium. Right. Now let's see if I can run a small video. Second. Okay. Right. Yes. So this. Okay. This small video I took from a book by Robert Weinberg about cell biology, cancer biology. Of course, you know that. So here you can see individual cells, and in green, cadherin is marked. So you can see how cadherin collects there where the cells are get connected, and then you need a lot of this cadherin to establish these connections. And also, I would say maybe. Probably it's clear to everyone in the room anyway, but so I would, you can realize it's a bit like this binding. I would imagine it's like if two people bind together by holding their hands. The only difference is, one of the differences I think is that cells, in order to establish stable junctions, they need a lot of these bindings. So between any two cells, you need a lot of bindings through cadherins in order to establish this connection. Right. Now I wanted to switch back to my presentation. Yes. Okay, then one more picture here. So again, this is taken from the same book. So here you can see now cadherins in brown, marked in brown, and you can see on the left. So now this is what one of the main applications we have in mind is, of course, modeling tumor. What happens in the tumor. So this is the case of a colon carcinoma, yeah. And on the left-hand side, you see a tumor core, and there's obviously a lot of cadherin present here. And on the right-hand side, you have the tumor edge of that same tumor, and you also see with this arrows marked some cells that already moved out of the tumor, and they seem to express less of this cadherin. So I know, as we already were told in one of the previous talks, that it's not only about cadherin, it's also a lot about tissue, but let's see if we for the moment concentrate on cadherin, and this is of course an important component, and we would like to include it into our modeling somehow. Right, now I will maybe dig a little into multiscale modeling, maybe even saw this picture before, I think shown by Christina. So you can model on different levels. You can model on the level of the whole tumor. This is macroscale. You can model on the mesoscopic level of many cells and tissue, or you could go even lower than that on the level of individual cells. And in general, depending on which level you model, you use different kinds of equations. So on the microscale, you use ordinary or stochastic differential equations. On the mesoscale, you use kinetic transport equations. This is high-dimensional integral PDEs. And on the macroscale, you can have either partial differential equations, or in more complicated case, you can have integral partial differential equations. Yeah. So. Which scale is sort of better? There's no simple answer, of course. It depends on what is more important for you for the moment. So eventually, the lower the scale, the more accurately you can model. But the price to pay is that it's difficult to analyze, both analytically or numerically. And also, there's a question of comparing with typical medical data. So macroscale is closer to what you usually get from such data as, for example, MRI. Yeah. So the multiscale approach allows to get the best of the both worlds, namely that you model on a lower scale and then perform one or two, if necessary, upscalings in order to get a macroscopic model. There you can do simulations and then potentially compare with data. And importantly, some lower level information that you had on the lower scale, say level individual cells, is still inherited on the macro scale. So this is why this approach, this is really, I would say, it works better as compared to when you would model directly on the level of tumor density, on the cell densities. Yeah. So our particular way of dealing with this and our particular adaptation, this approach is, it goes as follows. So we decompose, and this was also maybe mentioned in the talk by Andreas, that you have directed movement and you have also some stochastic driven movement, some stochastic perturbations, yeah? So what we do is we try to model as much as we can with simple means, ODs, not SDs, on the microscale. So we model all the directed part of the motion on the microscale, on the level of individual cells. And then, so we scale, so we use so-called empirical measures to get a mesoscopic equation, which is a transport equation. And in addition, and then the stochasticity is introduced on that level to get a kinetic transport equation. So we introduce the turning operator, yeah? And then we perform an upscaling to the macroscale, and we get, depending on the on our setting, we get either a PD or a reaction, diffusion taxis equation, or we get a non-local PD, for example, diffusion adhesion. So how does this work? So we use the usual settings. So the time we have variables, at least 3 variables: time, position, and velocity. Later I will add one more variable. This is this, so to say, the proportion of bound cadherins, but that will come later. And we denote on the mesoscopic level, on the level of distribution densities, we denote, for example, you have variable C by CT of XV, so depending on the 3 components. And on the macro scale, you use the macroscopic density, which comes out if you integrate this quantity with respect to velocity. So the scaling out to the macro scale means eliminating this dependence on velocity. And this integral will be denoted by bar. So we assume the velocity to be, we assume that velocity cannot go above a certain maximal possible speed. Yeah, so the speed can go above a certain value, and this value is scaled to 1. For which reason we assume that velocity lives in a unit ball centered at 0. Yeah, so all velocities are possible apart which have modulus smaller than 1. So now, the first effect that we include, we need to take care of 2 effects. One is this directed motion, and the other is this stochastic-driven velocity jump process. So I won't go much into that because this is now standard in this field, how you deal with this, what kind of turning operator you introduce. Again, we assume, and this is why we have still this application of into tumor in mind. So we have this, again, knowledge of stimulus following tissue fibers. So we model the turning probability by the fiber orientational distribution, but this is quite common here, so I won't say much about this. As to directed movement, this is built, this contain 2 components. On the one hand, we have a deceleration due to viscous force. And on the other hand, and this is the core part of this model, is realignment with a certain preferred direction, yeah, which we call V star. And it depends on time and position. The choices for this, and in most previous work, so if you know, for example, the paper by Hirin and Painter, 2013, the one where they discuss in much detail this kinetic transport equations with this turning operator of this sort, yeah, usually you don't have this directed component. Or on the other hand, if you look at models, for example, often used for models, for example, modeling self-propelling populations, such as for birds or fish and so on, then usually they concentrate more on the directed part of the motion, and they get that leading to loss of equations, for example. In our modeling, we have both effects. So how do we choose this sort of preferred velocity? So one case is that the one we looked earlier with Christina is when we want to model chemohaptotaxis, in which case you would expect that cell would want, apart from the stochastic perturbations, that cell would want to follow the gradient, so roughly speaking, the gradient of certain chemical. One, it could be either density of tissue, for example, leading to haptotaxis, or the concentration of some chemical leading to chemotaxis. So this gradient of the quantity Q. So we can also count for environmental heterogeneity. This is modeled by this F here. Yeah. So this is, but another way what you also could include is cell-cell adhesion. And cell-cell adhesion we describe here in the following way. We use a standard description. So this cell-cell adhesion comes through cell intracellular interaction between cells. And this kind of interaction is often described by interaction potentials. Yeah. So, and So here you see essentially the averaging sampling over these interactions with individual cells. Yeah, so say cell I interacts with all other cells, and the averaging effect will provide this direction. In addition to that, we also have, and this is, so if you think of these models that I mentioned previously about modeling self-propelling populations, usually they don't have this coefficient, this sensitivity to this binding, to this interaction, which we included here and which can account for example, environmental heterogeneity. So how, depending on the situation, maybe how much tissue there is or some other quantity, some other chemicals, maybe cell will be more keen to follow this direction or less. Yeah, so this is something you can have. So, and of course, what this kind of modeling allows, you can include both effects. So the one with taxes, so more effect of tissue, and the other is with adhesion. So I will concentrate on them. I will consider them separately, but you could, it's flexible, so you could include both. So this adhesion, so the gradient of this potential has a this distance-dependent component and direction-dependent component. So roughly speaking, it is this: if I have a cell sitting here and I have more cells here than here, it's more likely that my cell will be pulled to where there more cells exhibit this effect. Yeah, and this is the kind of thing that also will result from such a choice. So as we now move on to upscaling, we would need moments. So zero order moments, first order moments, second order moments. Again, this is quite standard. Only keep in mind that we are now integrating not over unit sphere, but on unit ball, but we can easily establish connections between the corresponding moments. Yeah, and we use the usual quantities here: mean fiber orientation, EQ, and autocorrelation matrix, D of Q. And so now, in a nutshell, how we proceed. So first, this is first the case of this taxis case. So we set up this an ODE system. Yeah, it's a second order ordinary system of ordinary differential equations. So for each cell, I have one and the same kind of equation. Difference being that in the initial conditions, of course, are different. So notice this is a second order system based on Newton's law. So one, this is the obvious equation connection between velocity and change in position. And this is, so if you look at the right-hand side now, this is this viscosity, the result of this viscosity force, and this is this preferred direction. So if you imagine if this preferred direction would be a constant, then obviously solving this ODE equation, you would see that velocity stabilizes to 1 over a times v of star, so it will follow this direction. But v star depends on t and x, so the dynamics is much more complicated. So what we do then, we use these two empirical measures, and then we get a transport equation. We send the number of cells to infinity. We retain the transport equation. We introduce the turning operator on the right-hand side. This is the only part which I not so much like in this modeling because it's not so clean. The stochastic part comes directly on the mesoscale. So we are not doing it on the microscale because otherwise we would have to use ASTs. And there's also the question whether you can do it with the second order system or you have to simplify to a first order. So we decided that it's well established that this kind of turning operator gives a reasonable result on the macroscale, so we include it on the mesoscale. So I show you just the case of parabolic scaling, but in the same fashion, the way we upscale here from meso to macro doesn't depend on whether you're using parabolic or hyperbolic scaling. It works in the same way in both cases. We're not using, those who mathematicians and may know this, we're not using Hilbert spaces. We're not making assumptions such as, for example, the zero order moment, zero order approximation is orthogonal to the first order correction. No such assumptions. We're not using Chapman-Engskog. So the approach really is streamlined so that it runs for both scalings in the same fashion. So first we get this mesoscopic equation. So this is the left-hand side is this transport part. It accounts for the directed part of the motion, and the right-hand side is this turning operator. Yeah. And then finally, as I said, using this approach of upscaling, we get an adhesion. We get a diffusion taxius equation where you have this standard which always arises with this kind of turning operator, myopic diffusion. And here you get taxis. So V star will give you the direction of the gradient, for example, of the attractor. So notice then, different from previous, most previous ways of deriving, for example, taxis, if you know, for example, papers with Christian Schmeiser and co-authors, in our case, the directed path of the movement comes from the transport path. Yeah, so it's not coming from a choice of turning operator. And it has, I think it's more realistic because this is a directed path of the motion, that's on the one hand. And on the other hand, when you start, if you only if you set up a complicated kernel, usually this affects what you can do mathematically. For example, often you have problems with doing parabolic scaling because the assumptions for parabolic scaling are not satisfied. Whereas in this situation, we don't have these limitations and we can run both parabolic and hyperbolic scalings. Yeah. In a similar fashion, we run it for the adhesion case. So the case of cell-cell interaction. So now we have this ODE system with this preferred, if you remember, this preferred velocity described by this interaction between the cells. Then we scale again the same fashion. Yeah, we get a mesoscopic kinetic transport equation, and notice that this is sort of a combination between this, I think you call it Vlasov equation, or when you have just the transport path with a non-locality in the transport, and you have another non-locality on the right-hand side, which is this coming from the kinetic, so from the turning operator. So it's, you sort of have double non-locality on the right-hand side and in this term, this divergence with respect to velocity. Yeah. So, and when you scale up, you get a model for, again, myopic diffusion and adhesion. And this R operator is the operator, if you know the papers, they're a well-known paper for macroscopic modeling, original macroscopic modeling of cell-cell adhesion by Armstrong, Painter, and Sherrod. They have introduced this operator, and this is just another way of writing this term. And I forgot to mention, this is convolution with respect to the spatial variable. So what you get is a diffusion adhesion model and derived through the multiscale approach. And this term, and in difference to the model by Armstrong, Painter, Sherrod, the diffusion is myopic, in general. So again, I want to stress that the adhesion term comes from the transport path, from the directed path of motion. And I think it's the way it should be, because some of our past research shows that if you, that in the limit, so here I forgot to mention there is this variable R here, which is this cell sensing radius. So this is essentially the radius of a ball where if I have if I look at it as a cell and look at the ball of the sensing radius, then this is where the cell can interact with other cells. Yeah. So we showed in some previous paper that if with Christina and Kevin Painter and Mari Eckert, that if one can prove under certain conditions, and when this R goes to 0, you recover taxis or diffusion model, depending on how you choose the parameters there. So my feeling is that adhesion must also come from the directed path of motion. Yeah, and this is what in the model happens. And notice that because these effects are clearly separated, this matrix doesn't arise here in the transport path. Yeah. Okay. We wanted to get a feeling of how myopic diffusion can affect, how myopic diffusion and adhesion would work together. So we ran some very basic simulations in one dimension. We made the following assumptions. So we assumed here that the diffusion coefficient is growing across the domain, which is just one-dimensional interval, 0, 6. So it's like shown in this graph, it's an increasing function, just linear. And here I wrote out the equation for the one-dimensional case, so what it would be, if for such diffusion, taking basic assumptions on the adhesion operator. So this integral term gives me adhesion, this integral term, Notice this diffusion here is written in divergence, more standard divergence form. So you might wonder, where is myopicity, so to say? Well, this myopicity just was separated in this additional effect. So essentially, if you write out this in one dimension, when you write out this myopic diffusion, you get two components, this divergence like in divergence form diffusion, and this additional transport. So this additional effect gives you transport to the left. So you have three motion effects, one kind of standard diffusion, this additional transport, and adhesion. So the question is how this works together. So we are going to change the parameters. And notice that so this parameter theta just tells me whether I have myopicity or not. If theta is 1, I have it is myopic diffusion. If theta is 0, I have just sort of normal diffusion in divergence form. So I'm going to change these parameters, these constants, and see what happens. So what happens? So here is the case. So all these pictures, they correspond to myopic diffusion case. So theta is equal 1. And we looked at 3 cases. You have either very weak diffusion or very strong diffusion or something in the middle. And keep in mind that diffusion, these 2 columns are for the case where we assume that there's no perturbation in diffusion, so it's just a constant diffusion. So symmetric picture. I forgot to mention the initial condition is constant. So, and this is simulated under no flux boundary conditions. So not periodic boundary conditions, no flux. So if they were not for adhesion and not for this no flux boundary conditions, you would retain a constant value. So if you assume, so you notice obviously the effect of diffusion. If you have a very weak diffusion, these pictures show development over space and time. So if it goes up, it shows how it develops over time. And the lighter the colors, the denser it is. So what you see here, what develops is 3, in this case, for instance, 3 aggregates, quite dense aggregates. If you have a strong diffusion, then you just develop 1 aggregate, and it also takes time to develop that altogether. This case, I think, is also interesting. It looks like there will be 3 aggregates developing, but eventually only 2 develop. And here, this is the situation where you have the diffusion is not constant. So you see, you have the diffusion is stronger on the left-hand side and weaker, on the right-hand side, weaker on the left-hand side. And this probably explains why we get this aggregate here on the left side of the domain. And also keep in mind that with these 3 effects, the only effect that is density dependent is adhesion. So if you already developed a tight enough aggregate, diffusion has no chance because the adhesion will dominate the scene. And then we wanted to compare the situation with and without myopicity. So you can, in the odd columns, you have the with myopicity, and in the even columns, you have no myopicity. So essentially, it means we kicked out this additional transport to the left. So what happens now? So my reading of it, I don't know how to explain this exactly, but my reading of it is that the myopicity holds the aggregates in place, because here you see when there is no myopicity, they keep drifting to the right. Why this is actually so, I don't know, but that seems to be the effect. So essentially, myopic diffusion are not so well understood mathematically yet in the first place, I would say. So this is also an interesting case. Here it seems that due to the lack of myopicity, we don't manage to get the density mostly to the left. And so the diffusion, that's where the aggregation has more chance because diffusion is weaker. And what happens essentially is that no aggregate develops in the first place. So it's just no aggregate here. Right. So how much time do I have now, if you know? 15 minutes. Oh, great. So I have enough time. Okay, great. Right. So now I want to, but that was just sort of a first try with getting this. So that essentially we tried, we wanted to see if using my multiscale modeling, we can get to the model by Armstrong Painter. And we saw, yes, we can. And we wanted to include now this effect of cell-cell binding through cadherins. So I will just tell you a bit about that. yeah, so here in this picture, you can see what happens actually in this binding. So more precisely, actually what's bind, as I'm understanding, is that these dimers of cadherins. So first the cadherin, we have two cadherins of a cell that bind together, make this dimer, and then two dimers of two different cells meet. And if there is calcium enough in the system, they can bind. Yeah, so here what you see is this extracellular parts. Yeah, right. And yeah, so what changes do we need to make to for them in the modeling? So we include the dependence on an additional variable y. This variable lies between 0 and 1. And this variable describes the proportion of bound cell adhesion molecules to total, to all cell adhesion molecules. Yeah. So the larger this number, the more of them are bound for a particular cell, and the less, of course, the less. So on the mesoscale level now, we have a quantity C, this distribution density, which depends on time, position, velocity, and this state. And now the macroscopic, again, quantity, the density depends, again, should depend on time and position. So this time, if we want to get this macroscopic quantity, we need to integrate with respect to V and Y. Yeah. So this is what I now denote by this bar here. So what changes do we make to our model? So remember, I showed you previously this V star, the sort of preferred velocity, the adhesion case. The difference now is we assume that the sensitivity to this binding interaction between the cells is proportional to this proportion of bound cell adhesion molecules, Yi. So it seems plausible. The more the bounds are, the stronger should be the effect. Since we have these additional variables, so for each cell now I have, for each ETH cell, I have now this Yi. So somehow I need to model that as well. So I need equations for that. So we assume sort of kind of something like more law of mass action. So we assume that if I have two unbound cell adhesion molecules for different cells, if they bind, we get two bound molecules for each. Yeah, so this how explains this reaction. So this binding and unbinding happens, it's reversible, so binding and unbinding. We assume here for simplicity that the total number of the cell adhesion molecules of a cell is constant, but this can be maybe, I think that could be changed. So but in that case here it is constant. And we need to finalize this modeling on the microscale. We also need to model this binding rates, and we assume that this binding rates K plus and K minus depend on the concentration of a chemical, so such as calcium, and the distance between the cells. So that's how we get this description. So S here is this chemical, for example, calcium. So if you put this together, yeah, an example would be, for example, cadherin by a cadherin dimer and concentration of calcium. So if you put this together, you get a complicated looking system. So these are the two previous equations for cell position and cell velocity, and then this third equation for this binding state. Now I know it looks kind of complicated, but if you just pay attention to the red terms, you will notice this is something typical you get from when you have a low mass action, you write the corresponding reactions, and this is the kind of terms you get. Yeah, it just looks a bit complicated because the rates are dependent on these different variables here. So if you use that same approach as I showed you, as I indicated previously, you get the following model, which I think very unusual, because on the one hand, you get again this diffusion adhesion model with myopic diffusion. And again, this is this adhesion term. But now, so C naught bar is the zero order approximation, the macroscopic part. Yeah. And it turns out that another variable arises through this upscaling, which is the first order moment with respect to variable y. So this is what I marked in blue color here. So since you have now two variables, one equation obviously is not enough. The additional equation that arises is this equation, where this new variable, this first order moment, is compared to the zero order moment, connected in the following fashion. This y is a rational function into which you plug integral operators. These operators arise from these binding rates. So essentially, you have one integral operator on the top, and on the bottom, you have sum of two integral operators. The integral operators are linear, but because you plug them in a rational function, you get a nonlinear equation. So you have a highly nonlinear, non-local system. So not just one equation with non-locality, but both. And this is a very non-standard equation because it's not a PDE, it's just a certain integral equation. Yeah. Currently, we are analyzing this equation, hope to complete the analysis soon. So I might say hard nut to crack because originally we thought, okay, linear integral operators, they're compact, nice operators, but here there's no compactness in time. So it turned out more complicated than we expected, considerably more complicated. So unfortunately, we don't have yet simulations for this model, so I cannot show you some nice pictures showing how it works out, but this is the work in progress. But again, I think the message, I think let me maybe, how much did I have? 5 minutes, okay. So I think what I want to spend these minutes on is maybe briefly compare this approach to what's been done previously, yeah? So what we found in the literature is the following 3 ways how one could derive this model. Yeah. So the first first method is when you start essentially with the first order SD system. So this is a situation where you would model everything on the microscope without this artificial introduction of the turning operator later. However, my understanding is that in this situation, you have to succumb to a first order system. So you don't have this like position direction derivative with velocity direction derivative with acceleration. No, you must have an equation directly for the derivative of x with respect to time is something depends on that, which depends on x. Yeah. So because otherwise you get strange terms on the mesoscopic level. So maybe you don't want that. And in general, dealing with SDs in this upscaling, you can do it rigorously. There are some works by Morale, Capasso, Ohlschläger. You can do it. It gets quite complicated. And as I said, the drawback is that you have this, you have to simplify on the microscale in that respect. But there's a lot of there are technical challenges because you deal with SDs. So the second approach we found, this is based on a paper by Budenschon, Hillen, Gerisch, and Painter. And this is when you develop these models using an integral master equation. Yeah, so this is not velocity jump process, but position jump process. And finally, there is a way, maybe closest to ours, where you have it based on you start with the kinetic transport equation. However, the whole modeling goes into the turning operator. So there's this additional directional part with this divergence, if you remember, I showed you with respect to velocity. So no, this prefers direction. Everything comes from the turning operator. And but that also allows to get adhesion. However, there is again if you follow this approach, I already said a few words earlier about that, you're bound in the sense that you can only do, for example, hyperbolic scaling. So you cannot do parabolic scaling, and you need to really choose the kernel in such a way that it gets you the effect you expect. Yeah. Also diffusion, which is tricky also when you deal with hyperbolic scaling, and and finally, maybe if we want to compare our model, our approach to the previous approaches, yeah. So we asked ourselves, how did it come about to adhesion term, to the diffusion term, and whether this comes dynamics was involved. So in our approach, what happens is that adhesion comes from deterministic part of the motion. This is, I think, the way it should be, and also through binding of cell adhesion molecules in that extended model, and the diffusion comes from the stochastic part. And the adhesion, so this binding also happens in deterministic fashion. Whereas, for example, in the approach 3, the adhesion term comes from the stochastic term, from the kinetic, from this turning operator. There's no diffusion and no comes binding. Yeah. This in the approach number 2, you have CAMS binding, but both adhesion and diffusion are coming from the stochastic motion, because you have this reproduction, how it's called, this kernel which you need to describe this in the space jump process in the master equation. Yeah, so everything comes from this kernel. So again, you have no directed movement. Yeah, yeah, and in the first method, the adhesion comes from deterministic fashion, in the deterministic way. The diffusion, you have both deterministic and stochastic effects, and no CAMS dynamics. And there is this limitation also, as I said, you have to succumb to first order system. Right. Yeah, and maybe I'll stop at this point. I won't rush into showing the scaling, and thank you for your attention. The talks are mostly built on the paper, on this recent preprint with Mabel, and an earlier paper which was for the taxes cases, the paper with Christina.